Hello, my name is Yasoni, and today we're going to talk about the effectiveness and reliability of UVC sterilization for surface pathogens at 253.7 nanometer fluorescent tubes versus 260 to 280 nanometer LEDs. I'm affiliated with Arizona State University with the Physics Department, Microdrop Diagnostics, SI2O2 Innovates, and UV1 Hygienics. Our outline is that the problem is a SARS-CoV-2 pandemic, which requires a need for safe, reliable, and fast disinfection. Our solution is UVC from 0.2 to 0.28 micrometers, which associates the DNA and RNA and kills pathogens. Our two sources are fluorescent tubes and LEDs. Our approach / quest is to find out how stable and reliable are UVC sources, and which one is better, fluorescent tubes or LEDs for sterilization. The methods we use were to map the power density rho for 3 fluorescent to 3 LED sources as a function of geometry over 3 months of use. Our protocol was to measure the power density of 3 identical fluorescent and LED sources 3 times for 60 seconds every 10 seconds for a total of 63 samples. We were to measure the pathogen kill rate in 2 minutes via AnovaBug and using pathogen cultures of lactobacillus and CFU counting. The results we found were that the LEDs had a power density that varied with a plus or minus of 6.25%. For 4 LEDs, the 0.25 millimeters squared, the kill rate of 99.99% at 1 centimeter. The fluorescent tubes had a power density that varied greater than plus or minus 15%, and a 10 centimeter squared tube with a kill rate of 99.99% at 1 centimeters as well. So the problem, yes, it's the SARS-CoV-2 pandemic and pandemics in general. Here's a 1500-year timeline of pandemics, with the oldest one being from 541 to 542 AD. And this is the death toll, with the bubonic plague having over 200 million deaths, and the SARS-CoV-2 pandemic or COVID-19 having 5 million and counting. With the Delta and the new variants going to increase this number. So this created a need for reliable, fast disinfection. So the most effective UV is from 200 to 280 nanometers, and they can kill these pandemics. So what is the minimum UVC power density that is required in 60 seconds for minimum energy for a kill rate that is greater of 90% or 99.99%? The answer is that for each bacteria, virus, fungus, there's a specific minimum energy required. So as you can see here, Aspergillium or mildew is a fungus and requires 397 millijoules per centimeter squared to achieve a kill rate of 90%. Compared to influenza or the bird's flu, which is only 2.3 millijoules per centimeter squared for a kill rate of 90%, is much higher. Now compare that to a bacteria, such as a Campylobacter, and this is only 1.6 millijoules per centimeter squared for a kill rate of 90%. And for coronavirus, it is a 23 millijoules per centimeter squared to achieve a kill rate of 90%. So it changes with each type of pathogen. So what controls a pathogen's energy threshold to reach a kill rate of 90% or 99%? The answer is that the energy increases with the number of nucleic acid pairs in the pathogen's genome. So the more resistant to UVC is from the antibiotic resistant E. coli, requiring 20 to 100 times more. So as you can see here in this graph with E. coli, the gene giving resistance to specific antibiotic in E. coli from MPHA, SUL1, tetracycline, BLA, TEM1, we can see that the computed energy at millijoules per centimeter squared to achieve a kill rate of 90% is much higher than the measured UVC energy for a kill rate of 90% on non-antibiotic resistant E. coli. For example, the tetracycline is 53 millijoules per centimeter squared for a kill rate of 90%, while it's only 2.6 for non-antibiotic resistant E. coli. Our approach was to find these key questions. Is the efficacy of the rapid 60 seconds disinfection that was critical, we want to find how to reach a minimum energy threshold and achieve a minimum kill rate that is greater than 99.99% for the pathogen of concern. We want to maintain a minimum power density of microwatts per centimeter squared for 60 seconds, the minimum energy, which was the power density times 60 seconds. So our two questions were, how stable and reliable is the power density in these UVC sources? And how does stability and reliability depend on the type of UVC sources? Our design of experiments, or DOE, was that we use two types of sources with 3 units each. With 3 identical fluorescent tubes, which were wands at 253.7 nanometers. They are FDA and EPA certified, and 3 solid state LED domes from 260 to 280 nanometers. They are more compact and use less power. So the key comparison was how effective are each at dissociating DNA and RNA and pathogens with the kill rate. Our method was that we had 2 types of UVC sources, and we want to compare the power density using a gallium nitride detector. As you can see here with this graph, the 253.7 nanometer fluorescent tube and 260 to 280 nanometer fluorescent LED, they both are in the UVC range for the highest effect, almost, with 254 to 265, so they are around that. And we use a BSL-2 sterile, clean room, full PPE, UV protection, and control geometry to find this. So some key issues that we had to comparison of the power density and power density stability is the nonlinear sensitivity of gallium nitride detectors. They differ for 253.7 and 260 to 280 nanometers, which were the fluorescent tube and the LEDs. And they conduct relative comparison of stability and are required to use 2 different detectors with different surface areas matching the UVC sources. Another problem was the aging of the gallium nitride detectors, and the fluorescent tubes switch on much slower than LED domes, which requiring a 2-minute cooldown for repeated measurements because of this. So comparing the 2 identical tubes of fluorescence with 253.7, our results are that there's a 37% difference between these 2, with the average power density of quartz tube 1 at 171 with a plus or minus of 8 microwatts per centimeter squared, compared to an average power density of 118 plus or minus 3 microwatts per centimeter squared for number 2. So comparing 2 UVC LED domes, we only see a 9% difference between the 2, with dome number 2 having an average power density of 71.57, the plus or minus of 2.51, while the dome number 3 had an average power density of 78.82, with the plus or minus of 8 microwatts per centimeter squared. We had a 2-minute switch up interval for each LED. So the results that we had with these 4 LEDs sterilization was the effective kill rate was different. So before UVC, we see a much greener image with the green fluorescence from DNA and RNA of the pathogen using cyber-safe green dye that we use on these slides. So after UVC, we see a much bluer image and less fluorescence from DNA and RNA of the pathogen. So the comparison we used was used by overlay and color intensity integration, as you can see here. So we found that there was a 30% decrease in green and a 175% increase in blue. And you can see that was 1 before UVC for the integration for the intensity of green, and the intensity of blue was 0.37 after UVC, which leads to a 99.99% kill rate. Our conclusions about the reliability of the solid state LEDs is much more than the fluorescent tube. After repeated use 1 month later, we found an 11% change in the power density for LEDs, while a 30 to 50% decrease in the fluorescent tube. We also found that stability of the LEDs varies from a plus or minus of 6.25%, while the fluorescent tube has a power density that varies with greater than or plus or minus 15% with the same parameters. This shows that the variability in solid state LEDs is much lower than the fluorescent quartz tube. The minimum energy varies by pathogen and antibiotic resistance, that's something we found, by 10 to 100. And this is due to differences in size, DNA, and antibiotic resistant genes. And You found that UVC sterilization via LEDs is a much more reliable form of pathogen sterilization in both bacteria and viral loads, minimizing pandemic spread. We found that it's one before UVC and 0.37 after UVC for the green and blue respectively, showing a 99.99% kill rate. Thank you so much.